Hi, welcome to this webinar about host cell protein analysis assay using mass spectrometry. My name is Thomas Kofod. I'm the co-founder and CEO of Alphalyse. I'm also a protein chemist working with mass spectrometry and protein and peptide analysis using mass spectrometry for the last 20 years. Today's webinar will very much focus on the assay we have developed here at Alphalyse over the last 6-7 years. We spent quite a lot of time developing this assay, and I want to go into detail with that. Applications will not be mentioned so much. If you want to look into applications of this assay, you should look at our website. We have other webinars about that. So first, I want to give you a short introduction to Alphalyse. We have been around for almost 20 years. We are situated with our main office and laboratory in Denmark, Odense, close to the local university, University of Southern Denmark, which has been quite famous for its proteomics research for many, many years. We don't do proteomics, but we use the same technology to look at proteins, especially biologics. And we have helped clients all over the world with the characterization mainly of biologics. So you can see here a list of the types of biologics we work with: proteins, of course, antibodies, antibody drug conjugates, biosimilars, vaccines, and also for the last 2 years a lot of gene therapy products. So as I already mentioned, our technology is mainly based on mass spectrometry. So over the years we have been involved in a lot of peptide mapping projects, identification of proteins, molecular weight determination, intact analysis, quantification, and also, of course, a lot of modifications looking at glycans and disulfide bridges. We actually discovered 7 years ago that a lot of our clients were using our analysis to look at host cell proteins. And when we looked into this area, we could see that mass spec should have a very big role in analyzing host cell proteins because it's much more detailed than the golden standard ELISA assays. So we started developing this assay that I will go through today 6 years ago. And we've been working on it since then, and for the last 2.5 years we've been selling this service, this as a service. So when we started out the development, we had a clear focus that we wanted to use mass spectrometry, of course, for identification and absolute quantification of individual host cell proteins. We also wanted to be as sensitive as ELISA, so we needed to have a sensitivity to the low ppm level. For us to be able to have this as a validated kind of method, we wanted to not only have the sensitivity comparable to ELISA, but we also needed the assay to be robust and reproducible. And this means that no matter what instrument we run it on, no matter who is the application chemist doing the analysis, and also over time, we should have the same data. If we run the same sample, we should get the same data. And it should be possible for our clients to actually compare results they got a year ago with the results they will get today. Otherwise, it wouldn't make sense to use it to monitor host cell proteins. So the assay is built up by a 3-step procedure. First, we have a sample preparation, and in here the sample preparation starts out with a sample. It can contain, it can be a dirty sample or it can be a purified drug product. We can do the analysis of all kinds of samples. The idea is that before we do anything, we actually add standard proteins to the mixture, so we know exactly how much we add, either 100, 500 or 2000 ppm of a standard mix of 7 proteins. That's represented here by the blue ones. And then we do a precipitation, so all the proteins are precipitated, and we can then remove a lot of the other stuff that might be in there, buffers, DNA, salt, and so forth. Once we have done this, we redissolve proteins and then do a digest. So we don't look at the proteins intact, but we digest the proteins into peptides. So we end up having a mixture of many, many peptides. And of course, we have a lot of peptides from the drug substance, but also a lot of peptides from host cell proteins. And this we then analyze by mass spec. The mass spec analysis is a standard mass spec analysis, you could say. It's an HPLC system in front of a mass spectrometer. The LC system is making use of a reverse phase column. So we load the mixture of peptides on a reverse phase column, and then we elute off peptides by using a gradient of acetonitrile into the mass spec instrument. And then the mass spec instrument is recording what it's seeing. Typically, when you run mass spec instruments and doing peptide mapping, you would run it in what we call a data-dependent acquisition mode. So this means that at any given time point, the MS instrument is scanning the MS1 to look at peptides. So it's identifying peptide by mass over charge. And then it's selecting individual peptides and doing MS-MS sequencing on each of those. It can typically detect and run 10 to 20 peptides in one round of analysis. This works quite well for most abundant peptides, but if you have less abundant, and those are the ones we typically have for host cell proteins, then the instrument might not select the same set of peptides. Even if you run the same sample twice, you might get two different data sets, especially with the low abundant peptides. And this is not very good when we need to have something that is robust and reliable. So instead of running in data-dependent acquisition mode, we have changed it to data-independent acquisition mode. This is what science call SWATH. And here we have divided the whole mass range into smaller windows, typically 50 to 100 mass windows. And then the MS instrument is doing MS-MS sequencing in each of these windows. And by doing this, you record pretty much every data. So you don't miss out on any peptides. You get MS-MS sequencing for each peptide that are in your sample. So if you run the sample 2 or 3 times, you would pretty much get the same data set. The disadvantage by this method is, of course, now you just don't have just a single peptide in each of the MS windows. You might have signals from more than one peptide. And this makes, of course, it makes it more difficult to read the data and do identification and quantification of individual peptides. We overcome that by building up an ion library that can search these more complicated data. I will come back to that. So that's actually how we build the ion library. Is built by data-dependent acquisition mode. So whenever we get a sample, we run it in triplicate in data-dependent acquisition mode to collect as much information about individual peptides and finding out how does these individual peptides fragment. And then we store this data in an ion library. And this ion library we use to search the SWATH data later on. Then when we do identification and quantification, we use not the MS1 signal, but the MS-MS signal, so the fragment patterns. And we typically have 6 transitions for each peptide we use to quantify. You can see them over here. You have 6 signal intensity from 6 transition of a single peptide. And then we use the sum of these intensities for all the peptides pointing at a certain host cell protein. So if you have 5 peptides pointing at a specific host cell proteins, we sum up the intensity of these peptide signals and then use that as a quantitative means of that host cell protein. We only do this if we have 2 or more peptides for a certain host cell protein. So we don't rely on any quantification based on just a single peptide. And then the signal is compared to the signal from the median response factor from one of the internal standard proteins we have added. I will show you that in a moment, how we do that. So to summarize the analysis, it's first an automated sample preparation. We actually managed to get the whole sample preparation into a single robotic system. So we don't have to do anything manually. This is also part of why our assay is so reproducible, because we don't have any manual pipetting steps or sample preparation steps. And as I just shown you, we started out by adding these intact protein standards, 7 of those, and then we do a precipitation and the digest and the peptide cleanup. And then following that, we run the sample first in triplicate in data-dependent acquisition mode for generating the ion library, and then we run the sample in triplicate again in SWATH mode. And finally, we can then read the data, identify the host cell proteins, and quantify them by using this sum-all method. This curve just shows you that by using this method, that we have good linearity. Here we have spiked in one of our standard proteins into a real sample from 2000 and down to 6 ppm. And as you can see, when we zoom in here, we have very good linearity all the way down to the low ppm level. Then most people will argue that knowing about mass spectrometry and proteins will say different proteins generate different peptides, and different peptides will behave differently in a mass spec instrument. And that's completely right. Different peptides some will fly better than others and generate a higher signal, even at the same concentration. And this is also what we see when we look at the signal from the 7 proteins we have added. We know exactly how much we added. We added 100 ppm of each of them, and they generate different signal intensities. You can see them here. If they were all on one slope, we didn't have any issues. We could just use that method to absolute quantify any host cell protein. But due to this differences in or variation of slopes, we have then decided that we always measure these in each sample and then use the median protein to quantify the unknown host cell proteins. So by doing this, we will sometimes overestimate and sometimes underestimate, but on average we will be spot on. So by doing this, we get a curve that we can use to quantify unknown host cell proteins. So if we have a readout of 500 for one of the host cell proteins, we can then go and read out how many ppm do we have of that specific host cell protein. We have verified the results we get from this absolute quantification using the sum-all approach by actually having a real sample where we were able to have 2 host cell proteins produced recombinantly, so that we could actually spike those 2 proteins into a sample, and thereby we will get this kind of a signal or slope, and we could then quantify what was in the original sample by reading out the point down here at the x-axis. And by doing this, we got for the PTS protein, we got 42 ppm, and for ferric uptake protein, we got 55 ppm. And then we compared that with the sum-all median response curve calculations, and those numbers are very similar, I would say. So this just shows that the method is reliable and also absolute compared to this other method of standard addition. We also show this on a E. coli cell lysate. So in this case, you could say we have an example where we have a sample without any drug substance. So when we look at the proteins we can identify and quantify, we are able to detect more than 1,000 proteins. And the most abundant one is at 62,000 ppm, and then we have all the way down to the low ppm level. When we sum up the ppms for each of these proteins, we end up at 1 million. And this is exactly what we should end up at since we don't have any drug substance in there. Typically, when you have a drug substance, then most of the signal will be from the drug substance peptides. But in this case, it's showing that we have an absolute quantification, and we can actually also see that it covers almost 5 orders of magnitude when we do this analysis. So, as I started saying, we have been running this assay for the last 2.5 years as a service, and we just passed more than 100 projects. And what we have learned from these many projects is that when we look at quantification, we are typically able to quantify down to the low ppm level, 1 to 20. It depends a bit on the complexity of the sample. We are able to identify sometimes below 1 ppm, and when I say identify, it's sometimes possible from a single peptide to actually identify a host cell protein. But remember, we have this criteria that we need to have at least 2 peptides pointing at any given host cell protein before we say we can quantify it. The precision is typically below 10%. We say to be certain that it's below 20. So that means if you compare results from different analyses and you could look at a specific host cell proteins, you can easily compare the signal or quantities from one sample to another. The accuracy, we have a saying that it's either double or half amount. That's the range where we actually can quantify, and this is due to these differences in slopes I just shown you previously. So if we record a HCP at 10 ppm, it could be 20 or it could be 5. The assay we developed from scratch within a few months, so 6 to 12 weeks typically. So, and this is something we have done on all the projects we run. We don't use data from individual or for other projects. We just base all the data on samples for each specific project. And the sample analysis, once we have set up the assay, is between 2 and 4 weeks. The method is can be used for all types of sample. We have used the exact same method for very, very dirty sample and very, very clean sample. So there's no differences whether it's from just after harvest or it's an antibody that is almost without any host cell proteins. And we have also used it for a broad range of different expression systems, E. coli, CHO, yeast, you name it. The only criteria you could say is that we need to be able to get access to a proteome database. That is a database of all the proteins that might originate from this organism. But most of these expression systems are related to E. coli, CHO, yeast. They are all fully sequenced. And this is then how a typical result would look like. In this case, we had a client that provided us samples from a 6-step purification process. And as you can see, you get the same numbers as you would get from an ELISA. So you have the total amount of host cell proteins down here. So you have almost 20% of host cell proteins just after step 1. That's just after harvest. And then when the purification takes place, we end up below or at around 400 ppm in the final drug substance. But what you also get, and this is the big difference between ELISA and mass spec, is that you now also get detailed information about individual host cell proteins. So you get the accession number, you get the name of the protein, and you get the amount of this specific host cell proteins throughout the purification, and you get the mass and the PI for that specific host cell protein. And this, of course, is information you can use for what we call data-driven process optimization. This slide is just to show you that there will typically be a difference between the results from an ELISA and the results from LC-MS. And here we have 7 different batches that have been analyzed by a generic ELISA with a platform ELISA and then with LC-MS. And as you can see, there's quite a big difference between especially the generic ELISA and the LC-MS data. And I think this is something we see quite often, that that ELISA is underestimating the amount of host cell proteins, especially in non-monoclonal antibodies. We typically have a higher level of host cell proteins than what you find from ELISA assays. But we have also seen the opposite way around. So this method, actually originally we were looking at using this as a replacement for ELISA, and I pretty much have to say that this is not going to happen, but I think it will be a method that will be used in combination with ELISA for many years. So it's very good to analyze and compare batches. Perhaps most of our clients are actually right now using this for bioprocess optimization, so it's a data-driven purification process. We also see that people are using it to look specifically at problematic HCP, because you now get this list of host cell proteins, so you can look, are any of those enzymatic, might be immunogenic. So by doing this risk evaluation, you can decide if you want to go back and redo some of your purification steps, or you are satisfied with what you see. We also have increasing number of residual proteins, so people are using different enzymes and other proteins during downstream processing, and of course we can easily look for those also and show that they are removed as part of the purification process. It's also very easy to compare biosimilar versus originator. As long as you can get hand in the originator, we can run the originator next to the biosimilar and show similarities and differences. And finally, we have also used the method to validate ELISA by looking at the coverage analysis. So instead of running 2D-gels and Western blotting, we can do the analysis of ELISA by using mass spec. So again, you get a list of proteins recognized by your ELISA and not just spots on a 2D-gel. So I think in summary, you should definitely use mass spectrometry whenever you start out a new biologics project, because you get access to a more data-driven process optimization. You can base your optimization on real data and not just guessing. You can very soon and early on in your project decide on problematic HCPs. You can make decisions if you want to remove those or if you are satisfied with what you see. You can control once you scale up the process, very easy to compare and keep an eye on what's going on while you are scaling up. And you can use it to select the best ELISA assay. So there's a lot of reason why you should use mass spectrometry as part of your biologics project. Finally, I want to say that we would very much like to discuss any HCP project you might have. We have a lot of knowledge from running more than 100 projects, so we would like to share that information with you, and you are most welcome to contact me directly by using my email down here, or you can also send an email to our general email account, info@alphalyse.com. And then I thank you for joining me for this webinar.